Tēnā koutou katoa. Welcome everybody. This is the second of our mammal pest talks for the Biosecurity Bonanza. It's my pleasure today to introduce Dr. Marcus Gronwald and Dr. Graham Hickling, filling in for Dr. Amy Whitehead, who's away today. Marcus and Graham are both wildlife ecologists based at our Manaaki Whenua Lincoln office. They've got collective interests in wild animal behaviour, invasive mammal monitoring, island ecology, as well as spatial prioritisation methods applied to wildlife management. Today's talk is on the topic of monitoring and prioritising management of deer and other wild animals, ungulates. There'll be a Q&A session that will follow the talk, so if you have questions, I'd encourage you to submit those in the chat during the presentation, and then we'll try and get through all of those at the end of their presentation. Any that we don't get through, we'll respond to by email. So just keep the questions coming during the talk, and we'll get to those at the end. So my pleasure now to hand over to Graham and Marcus to share their presentation.
Yeah, great. Yeah, thanks for the introduction, Andrew. I hope you can all see the presentation. And we will talk a little bit about deer management today. The first question that comes to mind is why would we manage deer? And there it is. There are two main reasons. One is quite obvious, it's the ecosystem health. So ungulates can do a lot of browsing damage and damage through grazing, as well as stripping bark, for example, to various New Zealand ecosystems. They do damage in tussock grasslands in the alpine areas, to pastures, agricultural sectors, as well as forests. On the left side, you can see one example what deer damage can look like. A healthy forest with lots of understory, seedlings growing, successive growth. And on the right side of the left picture, you can see quite a bare ground. So the impact of deer can be quite significant in these forest ecosystems, especially when numbers are growing. Another reason to manage deer is the animal health. In some areas, deer are seen as a valued species, especially for the hunting community. So for trophy hunting or meat hunting, you want to have a healthy animal and a healthy population. And on the right side, you can see in high density, there's competition for food and interest-specific competition, leading to a reduced health in the individuals. It's quite a skinny fallow deer on the right. But I've mentioned that we need to know kind of the numbers, like are they high or not, and how does that happen in New Zealand? Ungulate monitoring is part of the DOC tier monitoring. DOC tier monitoring has 3 tiers. DOC tier 1 monitoring is a very broad scale national monitoring scheme. There are around 1,400 sites in New Zealand that are monitored in a 5-year rotation, so it's 280 sites per year. And there's native as well as introduced flora and fauna that is being monitored. Monitoring ungulates happens through fecal pellet index surveys, which means there are lines being set up 150 meters long, and every 5 meters you look for fecal pellets in a 1 meter radius and count the pellets. And by this, you get a number of pellets per transect, and you assume that you have more animals. When you have more animals, you will find more pellets, and you can get an index of a relative abundance from these surveys. And when we look into what these results look like from the last 10 years, we can see that the trend for ungulate fecal pellet indices is going upwards. That means the populations are growing. One downside of this monitoring is that we're talking about ungulates, so we don't really know which species it is in this case. That requires a different kind of monitoring. But we can also see from these surveys that in 83% of the sites we found ungulates. So see that they're widely distributed, and there's a significant risk that they do quite some damage to our ecosystems. So how can we improve this especially of deer and maybe also goat? That's that's a question that we have to solve. And Doc wants to look into different kinds of monitoring methods. And Graham Hickling has done a review on these monitoring methods. And so I give over to Graham now.
Thanks, Marcus. So as Marcus just explained, there's a really long history of, excuse me, surveying deer with fecal pellet counts. And so my first year working for the Forest Service, 1981, this was a field trip up to the Harper Avoca recreational hunting area, and we were continuing what was probably a several decade long survey already in place then, and that has carried on to various degrees since then. So fecal pellet counting has been the fundamental of deer survey in New Zealand for decades. Technology has moved on a little since then. So next slide, please. And so last year, DOC's Monitoring and Evaluation Unit asked me to lead a review of what new advances in wild animal monitoring there might be and that would be useful for them. And this is wild animals in the sense of the Wild Animal Control Act. So that's the deer species, goats, tahr, chamois, and pigs. So not broader out into birds or invertebrates or whatever. So that's the focus of the review. It was a little bit of a daunting task, to be honest, because if you start surveying the literature on monitoring methods for deer and other ungulates, there is just thousands and thousands of papers. Now, it might have actually seemed easy because people have written books on how to count deer. The thing is, most of that literature is international, and a lot of it, there are just specifics of the New Zealand landscape that make monitoring particularly challenging here. You've got dense forest cover, you've got really rugged country that people can fall off if they're not careful. So a lot of the techniques that an American textbook might cover just really aren't suitable. Next slide, please. So I don't have time today to go into any of the techniques in detail. This slide is really just to make the point that there's a lot of different approaches to surveying deer and other ungulates. You can work from the ground, you can fly in a helicopter, you can fly a drone, you could look for the animals themselves, you could look for fecal pellets, you could look for sign, browse damage. And these days with modern technology, you can look for traces of DNA, environmental DNA. What I did in the review was go through each of these and look at sort of strengths and weaknesses and practicality for the New Zealand condition. Boiled that all down, next slide please, to a couple of decision trees. And again, I'm not going to go into the detail, but the point I want to make is there's one decision tree if your question is about the presence or absence of deer in a particular area. So if you were mapping the distribution, if you wanted to know if there was an incursion, if you wanted to know if you'd successfully eradicated off an island. So there's a range of techniques there, and you can see in the center of that, you've got environmental DNA, which is, it's not, if you already know that there's deer in a catchment, environmental DNA is not useful to you because it just reinforces that. But if you don't know whether deer are there, that's potentially a very powerful technique. The second decision tree, next slide, please, is if you're interested in the abundance and possibly even the population size of deer. And so again, you see there's a whole bunch of questions you would go through to try and sort of narrow down which of all those various techniques might be suitable given the particular catchments you're working in, the questions you're asking, and so on. I just want to focus on one, the camera trap index. When Department of Conservation asked me to do this review, they were particularly interested in techniques around using motion sensitive infrared capable trail cams, which is obviously a new technology since 1981, and one which has a lot of strengths to it. For example, knowing which species you're dealing with. It can be really tricky to tell apart different species fecal pellets, whereas if you've got a photo, you're in a much better position to recognize in areas where there's maybe a mix of several deer species, maybe some goats and maybe some chamois. So the camera trap index, next slide, is something that we have tried under New Zealand conditions. There's a photo here taken by Grant Morris in the Timaru Creek Reserve down in Otago, trail camera mounted to a waratah. That was used to monitor trends in deer populations and tahr populations over a period of time. The index that we focused on in that decision tree is the most simple-minded approach you could use for camera data. It's basically saying, how many animals did we see for X number of cameras out for Y number of days? So, you know, cameras per 100 trap camera night or whatever. And so if you think about that, really what that is a measure of activity. So the more animals move around, the more photos will be taken. And so there's some advantages of camera trapping. There's some disadvantages in that anything that affects movement, you know, season, visibility, weather, all that kind of thing will have effects on the result. So camera trapping is an index, and in addition to that kind of basic approach to camera data, there's a lot of, as I did the literature review, a lot of fancier techniques that use more advanced statistics, more advanced methodology. Those haven't been tried in New Zealand, and so I'm going to pass back to Marcus because work that he's got just getting underway. Oops, I'm one ahead. What I was going to say also is, as recommendations to DOC, we came up with a draft specification. Peter Sweetapple wrote this on how you would do a standardized camera trap index. So that's been recommended as part of the DOC toolbox. It's not there yet, but they're going to take those recommendations on board, hopefully. But as I was saying, beyond the basic index approach, there are other methods. And so I'm going to pass back to Marcus now, and he's going to tell you a bit more about that. Thank you.
Yeah, thanks, Graham. Yeah, one other measure of abundance that I want to introduce now is density. So we've already heard about relative abundance, and Graham has mentioned some disadvantages. For example, that the indices are more a change in activity over time rather than abundance. However, these indices can correlate with true abundance. That has been shown for other species, not for deer yet that I'm aware of. But more activity can be caused by more animals. It's just not that easy to see. So density would be a much nicer measure, because density, usually expressed in animals per hectare or animals per square kilometer, informs you about the actual population size. So when I know there are 10 deer per square kilometers and I have 10 square kilometers, then I know there are 100 deer. This can help us making like really clear and precise goals where we say, okay, we want to reduce the population to 300 individuals, and we know through the estimate how many there are, so we know how many more do we have to remove, or can we stop intervening at a certain point in time. If we pair these density estimates with biodiversity monitoring data, then we can also develop density impact functions and see how low do we have to go with the population before we can get positive biodiversity gains, for example. There are different ways of estimating population density, and I want to introduce you to one concept, and that's the capture mark recapture concept. We have a population in a forest here. You can see the deer running around in the forest happily. So we go in and capture a few animals. In this case, we capture 10 animals, and we all mark them with maybe some spray paint on the back and release them again. So we have marked individuals in the population, and they mix with the whole population. Then we come again and do a second trapping session. And in this case, we caught 8 individuals in the red box, and we see that 3 individuals are marked. So what we assume whenever we trap is that we get a sample that represents the population well. That means the ratio between marked and unmarked animals in the second sample should represent the ratio of marked to unmarked animals in the whole population. So we use these information to then estimate how many animals there are, because we already know how many we have marked before. And In this case, for example, the first equation shows the ratio that I just mentioned. The number of marked deer in the population should be similar to the number of marked deer in the second sample that we have trapped. Here they are called recaptures. And when we then rearrange the equation to population size, then we can calculate given we have the known number of marked deer and the known number of recaptures as well as the numbers of the second session. We get a population estimate of 27 deer. You all have counted 28 deer, I guess, so we are very close. The issues that we see and the challenges that we see with this method is that it's expensive. We have to go into the area twice. We need to do trapping. It's complicated. It's difficult. We need to handle animals. It's very invasive. It can have effects on the behavior of the animals. Some animals, if you give them a reward, they come back more often than a normal animal or the average animal. Other animals might become trap shy and just try to avoid us because they had a negative experience. So you change the behavior in the population. So it's quite a difficult method. And to really get the best out of these models, when we estimate density, we don't want just have a marked animal. We want to know which individual it is. We don't give them names, but we can give them numbers, for example, in ear tags, collars, microchips, or maybe dye the fur so we can identify the individuals. That brings us to the project that we're running with Andrew Gormley at the moment. And this is trying to figure out non-invasive methods to estimate the density of deer. And one method was already mentioned in the overview of methods is the DNA from fecal pellets. It's possible to identify individuals with genetic tools from collecting fecal pellets. So no animal handling is required. You can go into the area and collect the pellets. And then we can use these capture mark recapture models or capture mark recite models to estimate the density. That's the hope that we have. There are always challenges, and that's what is the error that we have when sampling? What are the costs of this sampling? We don't know yet, so I'm looking forward to seeing these results. The second method is using trail cameras. We know that trail cameras are used widely nowadays and they become cheaper, and there are certain protocols already in place for other species. Why not use it for deer too? One issue is that we can't really identify individuals well on camera. We can identify the species, but not the individual, unless it may be a male with a special antler that we can identify. It's always easy to set cameras and to collect data, but it's very time consuming to analyze them. So it might be a cost issue too. However, there are some new models that don't require individual ID of species. They're not perfect yet. They're not all tested in the field, and they have been tested on some ungulates in more open areas in Africa where the estimates were okay, but it's really hard to parameterize these models, and we need some additional data about movement. But trail cameras can also give us information about movement. So this will be a test of different statistic models that we can use from camera data. The good byproduct from both of these methods is that we will have a fecal pellet count, which is currently the standard method. We will have a trail camera index for relative abundance. So we can see if they at least correlate. If not, there must be an issue with one of these two methods that we haven't seen yet. If they correlate, maybe they're both suitable, and then we can choose the best method to monitor. So as you can see, the aim of the project is to find good tools to monitor deer, calibrate the indices. If we just stay with indices, maybe we will be able to get density estimates. The main goal is to make it affordable and feasible and have a non-invasive method that can then be transferred to other ungulates too. And all these fancy measures that we do are to inform management, and Graham will talk a little bit more about the management side in deer and how to prioritize it spatially.
Thanks, Marcus. This seminar was originally advertised as being partly given by Amy Whitehead, and she was going to talk about spatial conservation planning tools in the context of managing deer and other ungulates. And unfortunately, Amy couldn't be here today, so I've kind of stepped into the breach. But I just wanted to basically give you a teaser for a talk that she will give at some point in the future, which is, next slide, please. Basically, just to introduce the idea of how spatial planning tools could be used to address things like how we should manage ungulates in New Zealand. The issue in New Zealand is there's a huge range of opinions and values about how ungulate management, how deer management should proceed. And there's never going to be a one-size-fits-all solution that can be applied nationwide. So the solution is going to lie in different management regimes in different places. And we already recognize that. We have national parks versus recreational hunting areas. I was in a recreational hunt area in 1981. But we can apply these new tools to those kinds of questions of developing tailored management plans for different areas. And next slide, please. And the way that those kind of spatial models work is you are somewhat GIS based. You build up feature layers. Next slide. And so, the kind of data that would go into a spatial model, you would have things like the distribution of the ungulate species, which is what those maps show. You would have the vegetation that underlies that. You would have the management that existing plans implement, what DOC's doing, what hunters are doing, wild animal recovery operations, iwi, and so on. You have the other management that's going on for other ecosystem values. You've got underlying layers to do with land tenure, designations like recreational hunting areas. You've got a whole bunch of non-spatial data about other ecosystem threats and so on. And so these are powerful data-hungry tools that are available and have been used for things like reserve prioritization, but have not really been used in the context of ungulate management, invasive species management in the New Zealand context. So we're really excited to have Amy. She's recently joined the Wildlife Ecology and Management team, and we will be back at some future point for her to give a presentation on the work she's doing. Thanks, Marcus.
Thank you.
Excellent. Thank you both, Marcus and Graham. Any questions that you've got, put in the chat. Just to let you know, there was a first post that got put in there about an AI transcript of this session. I don't know if that's legitimate, so perhaps don't click on that link. Just a comment from Joris. Auckland Uni working on re-identification of stoats and deer and cats using a machine learning AI style method from camera traps. Would you, either of you, care to comment on how advanced that might be?
Well, I'll make one comment, which is that in the review that I talked about, we did identify the possibility of under New Zealand conditions, looking at to what extent we could identify individuals, particularly the males, because there is a study out of Scandinavia that took the approach that the antlers and so on were identifiable in the male animals, and then you could get the sex ratio of males to females and extrapolate up. So that was kind of flagged as a potential recommendation to look at the feasibility of, but I know Marcus has some thoughts on that.
Yeah, so I'm already collaborating on this stoat project with Professor Ko at Auckland Uni. And I know they are very interested in trying it on larger animals because it seems to be a bit easier on larger animals. I would think that they have more features than a stoat to be identifiable by AI. So it's certainly one option that is still part of this project that we have. If there's enough good data and time available, we will collaborate with them. One issue is that you need an extra data set to train the AI. So you need to know first which individual you have in front of you to tell or to test if the AI is correct, and that requires an extra layer of data collection. I'm not sure if this will fall into this project, but I'm closely collaborating with them, and I think it's promising, but it's always promising like all AI. So we will see how the test results will be.
Thank you. A question from Louise. Sorry, I've just gone around the capture mark recapture. What's the feasibility of marking deer with paint guns as an easier method for doing batch marking, if you like, for estimating rather than a catch and release? Just a thought there.
I would assume that it's possible to mark the animal. The question is, how do you change the behavior, and will you be able to recite or recapture this animal? Because, I mean, a paint gun is quite painful. And getting hit by that, it will be quite cautious, and it will have a negative experience on top of animal ethic concerns that might occur when you use this method.
Yep. I think I'd also make the point, Andrew, if you look at that photo that's on the screen right now, the practicality of large area survey using a method like that, it's fine for like a small specific catchment. But particularly for Department of Conservation, they're dealing with vast areas, and so there's a lot of method. Like every method would probably work if you put enough money towards it, but that's always a really limiting factor. And so those intensive techniques are really more useful in a research context than in a sort of a national management context.
Good point.
I also want to add one more thing, because we have talked about invasive versus non-invasive, and the advantage of these non-invasive methods, like using the fecal pellets or the cameras, is that the nearly undetectable animals become detectable with these methods.
A comment from Marion. The Fiordland Wapiti Foundation seem to have a successful management program in place. Do you know what methods they're using for monitoring their stocks and population size?
I don't know which method they use. No,
yeah, Amy would be the perfect person to answer that. Let's just pass on that one, and I'll get Amy to respond on the.
Dallas from Osprey asking about what data is currently available to estimate deer population and densities across the country, because that's obviously something that they're very interested in from the TB perspective in terms of potential for deer-to-deer TB transmission and spillover. So yeah, I guess it's the wider question about what's the state of the current data nationally.
Well, I'll go first. I mean, there are national maps of where deer are present. There's crude estimates nationally of the relative abundance. It's a changing picture, though. We know that deer numbers are increasing. We know that some of the species that have been less common in the past, like sika and fallow, are changing distributions. And so that's really what's motivated Department of Conservation asking for the review. It's motivated the projects that Marcus and you, Andrew, have got started. So that how is the question worded? I mean, the question is, we don't really have good data at the moment beyond the distribution maps, and we definitely need to do better.
A good question here. Sorry, you go, Marcus.
Just to add to that is that when you want to estimate density, you need data, especially for this purpose, usually. So that was never really considered. Like the dog tier monitoring showed, there will be indices of relative abundance. What would be helpful is if we can show strong correlations between these relative abundances and density, so that we could make inferences, but the data to really estimate them, I don't think it exists.
George Ledgar from DOC asking a good question here about, so the density estimation methods, I mean, they'll have different levels of reliability, uncertainty around them, and generally the use cases will often be at where the ungulates are at low densities. That's where they're interested in about, the regeneration of preferred species occurs at low densities. So they're interested in a reasonable level of accuracy at these low densities, less than 2 deer per square kilometre. Do you have a sense of which of the estimation methods would appear to be the most suitable for that type of a use case?
So the highest precision or the smallest confidence intervals you will get with spatially explicit capture, recapture modeling, and I would see the fecal DNA pellets as maybe the most promising tool, because it will give you the location of your sample. So you will have a GPS coordinate of your sample, and then an individual ID. If that works well, then we have got data for quite robust estimates. But again, it's one thing that we have to test first before we can make promises.
And just a comment on fecal DNA. One of the challenges is how fresh does the poop need to be, which is kind of a funny question to be thinking about. But we heard a presentation recently from Australia where they were saying they had a relatively short period of the DNA staying viable enough to do individual ID. And so you get into this guessing game of like, there's a nice glistening, shining fecal pellet, but maybe it just was rain or dew. And so a lot of these methods, it's testing how well they work in practice under New Zealand conditions.
We're slightly over time, so we've got one more question here, so we'll address that. And then if you've got any other questions, please send them through, and we can respond to those in time. Question here: if either of you can or would care to comment on what this government's appetite is to further ungulate monitoring, or WAC, which I think might mean wild animal control, across these species. So where does the government sit in terms of wild ungulates?
Well, we don't know at all, but I would just make the comment that we now have a game minister, which is a new development. I think there's increasing interest from this government in herds of special interest. Along with managing a herd like that, you would need decent monitoring techniques. So in terms of funding for research and funding for more monitoring, who knows? But there's certainly, I think, a growing recognition across government that rising deer populations is something that probably needs some more thought, at least.
Excellent. All right, thank you very much. Thank you both to Marcus and Graham, and to Amy for supplying some material for that as well. And appreciate everyone's attendance and the insightful questions and comments. So this afternoon we have Dr. Simon Fowler presenting an overall cost-benefit analysis for weed biocontrol in New Zealand, and from the WEM team, Wildlife Ecology Management team, join us tomorrow at 10:30 for Patrick Garvey, who will be talking around understanding and targeting survivors of pest control operations. So yeah, thank you all very much, and we'll see you at the next session.
Thanks,
Thank
Andrew.
you.